欢迎收看《家国纵横》，我是康凯。脱氧核糖核酸 DNA 的技术应用已经非常的广泛，比如1公升海水里鱼类的 DNA 不但可以探索到各种鱼类的存在，还能算出这些鱼类的相对数量。而空气当中原来也可以收集到 DNA， 从中获得很多的信息，包括有哪些物种在这些环境内生存，甚至有可能追溯到远古生物的痕迹。科学家是如何收集这些信息？知道了有哪些物种对环境保育可以带来什么样的好处？由于动物同环境互动时总会脱落一些基因的痕迹，近年常用的环境基因辨析方法，利用水样本也可以找到有关水域的生态。只不过空气是包围着陆地上所有东西，因此寻找空气中的环境基因，可更有效地找到陆地或水陆两栖动物的物种。
There have been some reports of people collecting dust from the air and using that particularly to look for plant material. That's been done for a couple of years. There's also a community of people who use air sampling to look for pathogens like bacteria and viruses and allergens. There has been almost no work looking for things like mammals and birds, and that's what interests us. I'm a mammologist by training, and we're looking at ways that we can add more tools to our toolbox that we use to survey for terrestrial diversity and there are some attempts that are ongoing right now to try and get insect DNA from the air. So I think this is a field that's going to grow very rapidly as people test out the methods in different ways. We still haven't quite narrowed down the best way to collect the material. We're looking at different ways of filtering large volumes of air, large amounts of sampling.
一个跨国度的科学研究发现，仅凭空气中的脱氧核糖核酸 DNA 残留痕迹，便可以推断到附近环境中曾经或正在存活着哪些物种。这一项发现有助于生物学、环保项目、考古学等多个范畴的科学研究，堪称脊椎动物生物监测的一项创举。
I think probably the environmental DNA carried in water is easier to work with. And there's a very long history of that being done. Lots of people work with aquatic eDNA. That's a really common substance to use. It's used by regulators and commercial companies, by governments, by researchers. It's a common source of environmental DNA. What we were really trying to do was figure out how we could take that technology and move it on land. On land, we've gotten environmental DNA from things like rain and snow, soils. Honey carries environmental DNA. People have even been spraying the surface of leaves and collecting the runoff to see if they can find DNA that's basically washed off surfaces. So there's lots and lots of sources of this material, but air is a new one, and it might be a little bit easier than some of the other terrestrial sources to collect.
除了水样本，空气样本也可以收集到 DNA 的信息。Dr. Calera 做了有关的研究，2份报告最新发表在今年1月初的科学杂志《现代生物》上，指出这是一种崭新式的非侵入性检测方法，借以监察生物多样性的记录及演变。
What we're trying to capture is very, very tiny amounts of DNA that seem to be floating in the air. And we use this to try and concentrate them. So if we're trying to take a large volume of air through a very small filter so that we try and collect as much DNA as we can, because it's very, very dispersed in the air. There's not much of it. We have to move quite a lot of air through one of these. We filter for more than half an hour to collect enough traces of DNA that we were able to identify animals.
参与研究的有2支团队，其一是以丹麦为基地，另一队是以 Dr. Calera 为首的英国及本国加拿大为主，分别在英国距离首都伦敦北面大约2小时车程的汉默顿动物园，以及位于丹麦首都哥本哈根的哥本哈根动物园。
We use a very small device like this. It's basically a very tiny filter inside a little plastic capsule. We normally use these for filtering water to remove particles from water, but it just pulls air through one end, out the other using a very low power pump. And anything that's in the air that's a particulate bit of material gets trapped on the filter. And then we can quite literally crack them open in our lab and remove that piece of filter, and we can extract whatever particulate matter has been caught from the filter. It's kind of like making coffee. You strain the water through and you catch the coffee grounds. we're straining the air through, and we're looking to try and catch environmental DNA on our filter.
当中，英国方面的研究是以本国约克大学助理教授 Dr. Elizabeth Clara 为主导。他利用真空吸管的具敏感性能的过滤网，收集空气中多达70个样本。这些浮游在空气中的环境基因，也可以被用来检测陆地上生存的动物物种。意外的发现，不单在动物园范围内，甚至在动物园以外的很多种动物物种。
My main area of research is I'm a bat biologist. I study bat diversity. And feeding interactions. So we study what plants have their seeds dispersed by bats and what insects they consume. So I've spent a lot of my life in different environments, catching bats and counting them and identifying them. Members of my research team also work on other groups like insects and fish, amphibians, birds. So we are really quite interested in the diversity of life. And these genetic approaches to trying to identify species give us really good information about what's in an area. And we're looking for more and more ways to collect material so that we can more rapidly identify what lives in this ecosystem and what changes over time. So if some animals are coming and going, we want to know that that change is happening.
Dr. Clara 指出，之所以是两组团队进行研究，完全是巧合。实际上，他与另一团队的组长 Christine Bowman 早已认识。当他们把论文放上网络后，就被告知另一丹麦的团队也做了同样的研究。由于两个实验非常的相似，而且科学在于不断的检证，于是两个团队同意保持独立研究，也不竞赛。开始阶段不互通研究信息，最终才发现两队的研究结果是惊人的吻合。
Our team identified 25 different species of mammal and bird. Most of them were from the zoo. We found tigers and lemurs and dingoes. And some of the primates that were residents of the zoo. We also found hedgehogs, which are endangered in the UK. They're vulnerable to loss of habitat. So we were really excited to find them. And they're not part of the zoo. They're actually part of the natural environment. The other study found a different selection of animals because their zoo has a different fauna. They had a lot of larger mammals that we didn't have. They also found guppies and reptiles, birds. We found some of the cranes that they had on the zoo, the birds there, and zebra finches and budgies. So we were actually able to get a wide variety of different species of mammal and bird, and some evidence of fish and reptiles in our sampling.
这项研究从一开始为了迁就两地不同团队的进度，所以并不在于快速完成，尽量做到同步进行。研究初期是各自行动，利用各自的方法进行，彼此都不知道对方的信息。Dr. Clara 再三强调，是直到研究结束，双方才惊叹实验结果是如此的接近。
Well, that was a big surprise. We didn't know about the other team when we were doing our experiment, and they used a very different form of air sampling filter. They had 3 different devices that they were testing out. We used this device because we'd used it before in a lab to actually test the system prior to. But the big surprise for us was that there was another team, and we actually see this as a big advantage because it means that we've replicated the study independently with no knowledge of each other and found the same thing. In both cases, the teams were actually surprised how efficient we were at collecting animal DNA. And we did identify the zoo animals that we were intending to try and capture DNA from. But we were also able to identify things like animals that are actually native to the habitat that weren't zoo species. So we found evidence of squirrels and ducks and hedgehogs, which were native to our area. And similarly, the other team found squirrels and some of the birds that were in the same location.
环境 DNA 主要是从环境内的生物剥落下来的毛皮，有些可能是不久之前，几个小时或几天。不过，单凭这些蛛丝马迹，还可以重组远古生态环境。
It can persist for long periods of time. In fact, the first environmental DNA was actually found in permafrost, about 20,000 years old. So that's the very first traces about 20 years ago of this ancient DNA in the environment. So those ancient samples are probably rare, and they're going to be very specific environments like ice cores where you would get them. More likely what we're going to get is DNA that is recent. And so its real application will be in things like biomonitoring for ecosystems now.
环境 DNA 的难能之处还在于揭示到了稀有物种，它们原本是从别的地区进入这一环境，还有了解到濒危物种的存在、数量及活动范围，更有助于生物监测等环境保育工程。不过，目前最大的挑战是过程当中的污染。
We don't know how quickly it accumulates in nature, and we also don't know how quickly it degrades in nature. So we need to understand how long the signal hangs around for. We also don't know how far it goes. We know from our experiment in the zoo that it can go hundreds of meters in air currents, but we really don't know what the maximum is. So it may be traveling very long distances, and we need to know that before we understand how to use the information. But contamination is also a problem. So everything leaves around these traces of DNA, and it's really hard to have a sterile environment where you can actually work with the samples. This will be a quite significant challenge. We see contamination from all different sources within our data. Just being inside a lab environment, you're exposed to all the DNA that's accumulated over decades of use of a lab room, including people and plants and microbes and things that just accumulate in our airspace. And so these can be contaminants that we have to learn how to filter out. Air seems to have all of this material in it all the time. And that's going to be a challenge for anybody who tries to use the method. And so what we really want to be able to do is to use this as a new tool to try and detect some of these very rare forms of life that may still be in our natural environments.
这种从空气收集 DNA 的研究方式大大缩减了研究的时间与人员需要。团队不需要长途跋涉、攀山涉水、冒险到一些偏僻地点，例如洞穴或深渊，也不需要肉眼见到这些物种。即使空气中残留的微乎其微的基因样本，便足以探索到研究环境内物种及其相关的信息。证据真是凭空而出，不过研究结果却是真实无误。以上就是本期内容，各位观众如果想重温的话呢，可以登入城市电视网站，点击节目重温当中家国纵横的链接就可以了。我是康凯，下期节目再会。